to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 70th episode of the Weekly Bioanalysis, the official podcast of KCAS Bioanalytical and Biomarker services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is John Perkins and I've been in the bioanalytical LCMS space for over 29 years. I'm here with my co-host, Dominic Guarino.
Hello, my name is Dominic Guarino and I've been in the LBA and cell and gene therapy space for a little over 29 years now.
Dom and I are members of the growing scientific advisory team at KCS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCS's services. We are thrilled to have you listening to the 70th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you're joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics and we'd be happy to discuss something that you want us to cover. So again, we're fired up to have you here and we are looking forward to a fun episode today. To kick us off for podcast number 70, Dom is going to go over today's podcast topic a bit before we jump into the news and resources section. Dom.
Yeah, John, we have an exciting main topic today. We're going to be looking at, we're going to be discussing our autumn conference season. It's a jam-packed one. Without sharing too much detail, but we go to quite a few conferences this time of year, and we're looking forward to discussing them and the value that they bring to KCAS and in general to the scientific community. So back to you with Jet, John. Why don't we just jump right in with the first news article? An interesting one here, John. Tell us a little bit about synthetic antibiotics and what maybe some of their potential, something we kind of forgot about.
So yeah, this is the article is synthetic antibiotic shows potent efficacy against superbugs, and this is a topic we return to on a pretty regular basis. Our limited arsenal in antibiotics and the potential for increased resistance and how that limits our ability to deal with things in the long term. And this is a group from Duke University School of Medicine, where they've actually figured out a potential approach to deal with antibiotic resistance, where they developed a small molecule that successfully killed off a wide range of bacteria in tests in human and animal cells, and they moved to mice later on. But this is it's looking at gram-negative bacteria which are surrounded by an outer lipid membrane. That's gram-positive bacteria such as Listeria and Staphylococcus aureus don't have that, but things like Salmonella and E. coli are encased in this unique outer lipid membrane. So the lipid membrane today has made it difficult to treat gram-negative bacteria, but this actual small molecule is designed to target that lipid membrane, break that down, and then open up the bacteria to attack by the antibiotic. And this is a strategy that's been done in the past, but the trouble is that historic drugs have all had cardiovascular toxicity both in animals and in humans, so it's never really gone anywhere. So this group actually really designed the drug by very methodically in terms of what structure they wanted on it. They had a number of candidates, and then they identified their one compound called LPC233 that basically binds directly to an enzyme involved in membrane formation, and so it stops the formation process. It says the shape changes over time, so the binding outlives the bacteria itself and the very presence of the drug in the body. So it's pretty potent against a number of different bacterial strains. And I'll pass it back to you, Dominic.
Yeah, it's a fascinating read, John. I mean, an incredible amount of work goes into this type of research. And as you said, there's gram-negative and gram-positive. Gram-negative is naturally more resistant, so they kind of focused on that. And then this LPXC. I'm not that familiar with that, but that's the key enzyme that they've discovered. And you very eloquently kind of described how there are some drugs that have been reported to maybe block this enzyme, but they have like other side effects like cardiovascular toxicity, which, John, I certainly don't want that, right?
Mhm.
This seems to be a nice step forward. They did a tremendous amount of work, over 200 synthetic inhibitors before they landed on, as you said, LPC 233. I think in general, they showed a tremendous efficacy in mice, and maybe you'll tell us a little bit more about that. But I think the main point here is this is so refreshing, John, for me. Like, this is the type of work that kind of stopped for years, or at least in my, kind of, I don't know if "stop's" the right word, but this is it seems to be a rejuvenation of the post-COVID sort of science. And the Duke researchers poured work into this. This is fantastic news, John, because it's not going away. We still have antibiotics that work, but over time we're going to have better antibiotics. And this seems to be a breakthrough, John. I don't know. I'm so excited to learn more about how this can help, because none of us want to think about a world where antibiotics are not working well.
Yeah, we're in this weird space where we know we need antibiotics, but you'll regularly come across articles that say you can't make money making antibiotics or developing antibiotics. And we've had history over the last couple of years of companies who have got antibiotics approved into market, but then they went out of business because they weren't making any money. And large pharma haven't focused on it. So it really needs a fresh approach, something like this, to hopefully kickstart things. Because you're right, it isn't going to go away. This is something that we absolutely have to keep a focus on. But this weird imbalance that it's a huge need for us, but it's a huge need, but we're just not. There's not the amount of research being done that is necessary to protect us for the long term. Just talking about a couple of things they did in mice. They looked at drug resistant E. coli, and they had two different mouse models, 5 male, 5 female drugs, and they did this intravenously, where one group got intravenous LPC 233, the other got saline control. All the mice in the control group died of sepsis within 72 hours. In contrast, 90% of the mice treated with a low dose of LPC 233, 100% treated with a higher dose, survived for the rest of the 10-day observation period. They then looked at a urinary tract infection, dosing them orally. This was female mice. Again, UTI with drug-resistant E. coli, by the end of the 3-day study period, the bacteria in the kidneys of mice that received the drug were 1,000-fold lower than mice in the vehicle or control groups. The oral version of the drug eliminated the bacterial load entirely, suggesting that oral LPC 233 can be an effective therapeutic solution for UTI. And they also looked at toxicity, because obviously that's a concern based on history, and they found that the drug was tolerated by rats, with the only attributable side effects that whether the rats ate a bit less and lost a transient amount of waste. But then testing on beagles showed that there was no effect on heart rhythm or rate, nor did it change blood pressure. So it looks like this is, it has legs, and I think they formed a company to actually take this further. And looking for partners to hopefully, they started up the lead author is a guy called Pei Zhu, and they've started up a company called Valen Biotherapeutics to take it to the clinic. So we'll watch this space.
Yeah, no, it's they did a lot of work. I imagine they're getting ready for first in humans, or maybe there's one more animal study, but they did, as you mentioned, mice, rats, dogs, and then they did some cell lines as well.
Yeah, sure, absolutely.
Cells, which are important. So passing all the what we call sniff tests, right, John? It's got it's doing all the right things, and good luck to their startup. And certainly if they happen to be listening, we could help you out bioanalytically. I'll give a shameless
Yes,
plug.
absolutely. We can
I'll give a shameless plug.
send Kevin Burns to Valen Bio. Yeah, absolutely.
Kevin, if you're listening, Valen Bio is the name of that company. Now,
Yes.
John, we can, but great. This is the science that you and I kind of remember at least studying, and it's great to see it back. Fantastic news, just from I don't know. I'll sleep a little better at night knowing that we've got somebody researching these things, because it seems to have, as you said, it's been kind of like no money involved in it, and now it's kind of seems like it's coming back, and certainly great work done out of Duke, and good luck to them. So, all right, John.
And funnily enough, the next news item is actually linked, because this was another article where it's actually someone looking at a similar strategy but for antivirals. And it's a new strategy for antivirals blowing up viruses' protective membranes. So it's, they're looking at an antiviral strategy for a wide variety of pathogens, looking at the fatty membranes that protect many viruses, including the ones that cause COVID-19 and flu, leaving them vulnerable and able to sneak into cells. So this is a New York University group led by Kent Kirschenbaum. He said they're hoping to disrupt the membranes, poke holes in them, strip them off, and then it makes the viruses vulnerable and non-infectious. And this is they actually looked at their compounds are called peptoids. They're synthetic molecules, similar to naturally occurring antimicrobial peptides, but they actually changed some of the structure to actually enhance their ability to rip apart these membranes. And this was actually cell-based studies that they're looking at. But I'll pass it to you, and then I'll come and say some more.
Yeah, John, they dubbed them peptoids, right? These things.
Yes, exactly.
The Achilles heel of the cell, so to speak. I mean, just really not really my kind of, surprisingly, I've worked on some viral work, but doing these viral assays was never a passion of mine, John. I don't know if that surprises you, but these are funky little tests that they can do to look at egressing of viruses, and but to me, and I'll go on a little bit of a diatribe. I believe the evolution of immuno-oncology, the evolution of tumors, the evolution of cancer, it's all virally related, John. And so, the papilloma virus is known to cause cervical cancer and head and neck tumors. Mm-hmm. It is the etiology of the disease. It is the cause of the disease. Now, that hasn't translated into other cancer types, but I'm kind of the adage that, you've got a certain amount of viruses in your body that are kind of part of your flora. They're kind of naturally there. They do some things that are symbiotic to you. But to me, being virally infected, they're opportunistic, and I think they cause all sorts of cancers. And I believe every cancer has a virus in it. We just don't test for it.
True.
So with that, John, I guess that's my little kind of need for antivirals and why I think it's important. But maybe you can tell us more about.
Yeah, and so this, like I said, it's funny how you could see the mirror between these two articles. And this again. It's like the Kirschenbaum was looking for so basically you've got the issue with viruses that they mutate and they adapt to any treatment. And so for Kirschenbaum, he was looking for a target that was less likely to do that. So he's thinking because the viral membrane hasn't really changed that much. So he thought if we could attack the viral membrane, then you got a way in to get to the virus. So there are antimicrobial peptides which will disrupt these membranes, but they have an issue as drugs. They're easily metabolized by enzymes in the body, and it was also hard to get them to attack pathogen membranes and leave human ones unscathed. So that was but by collaborating with the Stanford chemist and study co-author Annalise Barron, she actually she made key chemical structures to nitrogens on synthetic peptoids rather than the carbon backbone of natural peptoids to make the molecules tougher. And then they actually found that a side effect of that was that the peptoids selectively targeted viral membranes over human ones by honing in on one particular fat molecule, phosphatidylserine, that's not normally displayed on the surface of human cells. So they had that differentiator and that ability then to attack. And so in terms of application, they tested 7 peptoids against 4 viruses: Zika, Rift Valley fever virus, chikungunya virus, and Coxsackie B3 virus. And so 3 of those have membranes. The Coxsackie B3 doesn't, and they found that the peptides were very good for attacking those first 3, but not for the Coxsackie. And so their long-term hope is that actually if they find a suitable candidate, it will be effective against multiple viruses, and like I say, give us an alternative plan of attack against them. So it really does. It's a very interesting area of work, and hopefully one that has legs to for the long term.
Yeah, well, the Stanford group said they kind of did an atomic bomb to the to the peptides there. It's an interesting read, John, in that they were able to hone in on phosphatidylserine, which is not found on the surface of human cells. So viruses are incredibly fascinating to study in that they actually put out decoy. They have transcripts in them to make certain proteins, especially like hepatitis and a couple others. They will get the body to produce like MHC decoy compounds, meaning your body, if it's all lock and key based. So if your adaptive immune cells, your innate immune cells, they're always surveying your epithelial cells. And if the epithelial cells have anything that's perturbed, meaning too high a level of MHC or too low a level of MHC, and that could be MHC class 1 or class 2, but typically it's class 1, that triggers an event. And so when a cell gets virally infected, it actually downregulates a lot of MHC, and it kind of indicates that something's wrong. Well, the virus will transcribe proteins to put decoy MHC up, and that's some of what's talked about. They don't go into that level of detail here, but that's the mechanism of resistance, John. Think about how clever that is.
Sure.
Think about the pressures of the immune system and all that. So what better thing than phosphatidylserine? That is ubiquitous within human cells, but apparently it's not on the surface of the cells, so making it, in particular, a fat molecule. So it's located in fat cells. So it makes it a great member. They were able to see the virus has that, and it's not something that's going to be on the surface of cells. I mean, an incredibly, some of these viruses I hadn't heard of, John. The Coxsackie virus. I don't know if I knew what that one is, but it is definitely like, this is, again, this unmet need type of stuff, going after the hardest stuff, right? Hats off to any of the scientists who are tackling the non-glamorous things like Rift Valley fever virus, right? These are just things we don't really worry about. Maybe Zika a little bit, but that's I think the spirit of the scientist that you want to see. It's like, tackling these really hard things, knowing that they have application across anything. And I don't know, John, it is another really cool story. I love the collaboration between, it looked like New York University and Stanford University, if I have
and actually they've started up. They've got a startup business on this, looking at called Maxwell Biosciences in Austin, Texas. So they're not out of the woods on this. It's like they've got a proof of concept, but they also have the issue that oral bioavailability of these drugs is poor, so they still have to figure out the delivery. That again is something we talk about frequently. So they're actually the plan for their first trial is to look at these as a nasal spray formulation.
Yeah, that's the easy part, right? But that is definitely the development piece.
Mm-hmm.
The fact that they've gotten to this point, identifying that phosphatidylserine, and then, I want to rephrase it. The Stanford chemist said, Annalise Baron, she said she solve the stability problem with an atomic shuffle. John, I didn't know what an atomic shuffle was, but boy, that's got to be innovative.
So it's a switch around, I suppose, of carbons, nitrogens,
So, but you're familiar with that? Like, is that
well,
something?
I'm filling in the gap. I'm inferring from what she said
Yeah,
what's going on. So I'm not
but clever,
I'm not a
right? Like,
major chemist. It's I think it's sort of substituting in the peptide backbone nitrogens for carbons is how
is it clever,
interpret
or am
it.
I making a bigger deal? Like, is it really a clever thing, or really?
I think it probably is.
Okay,
I
okay.
mean, it's not something I know much about. I suspect it is a really clever thing to do, and yeah, we'll see. I mean,
Yeah,
who knows what other people might read off this and where they might go. So it's like someone does something that to us seems clever or a small step, and then other people read it and think, but I can apply that
yeah.
elsewhere,
That's how it
and
works.
then that's what research is all about. So yeah, I
Yeah,
mean,
that led to the Nobel Prize in medicine, as you know. Maybe we'll save that for another podcast, but that just got announced. The
exactly,
two
yes.
yeah, but it's just those little things that,
Mhm.
it's. I love solving a problem, John, and that's the way to do it. Is just with science isn't these massive. Well, sometimes it is, but usually it's a subtle little flip, a subtle little re-engineering, a subtle little, oh, let's just do the reverse of that. Just the subtle little things that are like, oh, why are we putting this on there first? Well, that's just the way we've always done it. Has anyone tried putting this down first,
Yeah,
or
yeah.
whatever sort of configuration that can rearrange or solve your problem? So great stuff, John. I think we got one more news article.
Yes, yes. So I just to slowly close that one out. They're looking first clinical trial next 12 months to test its peptides as treatment for chronic sinus infections, followed by possible trials for long COVID and brain fungal infections. So again, this is that theory that if they get the right candidate, it can deal with a lot of
One point
different
on unmet needs, right,
viruses.
John?
Yes,
Those are
exactly.
those are
Yeah.
highly unmet needs. Because right now, if you have chronic sinus infection, long COVID, or brain fungal infections, there's not a lot the
Yeah,
physicians doing
sure.
for you.
So more power to them. Yes. So last we'll move on. This is one that's been out there for a while, and we last podcast there were some other things that I actually put ahead of this, but even though we didn't include it, I thought we're going to have to talk about it sometime. And this is CRISPR solves mystery of why cancer cells have extra chromosomes. This is a long known thing that cancers have extra chromosomes, but I don't think people necessarily knew what the extra genetic material was for. And this is a team at Yale. So humans typically have 23 pairs of chromosomes in their cells, and having extra is known as aneuploidy. I hope I'm pronouncing that correct because there's a word I've never met before. These extra chromosomes are used to boost tumor growth and could be targeted as a new pathway for cancer treatments, according to a study that led by Vishruth Girish, a student at Johns Hopkins School of Medicine, and Asad Lakhani, a postdoc at Cold Spring Harbor Laboratory, both of whom have worked in the lab of senior study author Jason Sheltzer. So let me just jump down. So, like I say, cancer cells have long been known to be aneuploid. They have these extra chromosomes, but this group actually used CRISPR to basically edit the chromosomes from cancer cells and then see what the effect of that was. So by chopping things out, did it impact the cancer, etc. And in one case where they looked at, they removed extra copies of chromosome 1, which is linked to disease progression, and they found that cancer cells lost their ability to form tumors. So they. So that this is like very early stuff, and for them this is proof of concept, and this hopefully will indicate where people can go in terms of addressing cancer in the long term.
Yeah, John, fascinating stuff, right? And you're saying the word right, but I don't enunciate the best. It is aneuploidy, and it's just meaning the tumor does that, or the cancer itself will have all sorts of rearranging of your genomic material, and they kind of give this blanket term to it. Is how I kind of think of it. And so what they've been able to unlock here is if you can undo that, then you cure the cancer cell will die, right? And I just, it's like, as I sit here and look at this, I think back to like 1994 when I first started doing autologous T cells. And at the time, it was technically a gene therapy. And here we are looking at CRISPR all these years later, and they're able to do it at such a targeted level. It's like a wave of emotion coming over me around how far science has come, and couldn't be more thrilled to see this type of technology starting to really kind of unlock some of what we've known for years, right? They knew cancer cells had different, let's call them mitosis and meiosis patterns, right? All sorts of different replication, the whole concept of your P53 and your retinoblastoma genes. Those are like the brake is cut and the gas pedal's on. All of that plays into what is being described here. And every tumor cell, every cancer cell, every mutated cell kind of goes through this thing. And so the fact that they're able to restore it, right? They're calling it restoring disomy, right? Or redact is that it's just, John, it seems like Star Wars-y or Star Trek-y to me, but still a long way to go.
Oh yeah.
But the fact that they're starting to unlock it at any level just means in another 20 or 30 years there'll be something that's on the market for it. That's how CAR T cells got to where they are today. It's great stuff, John. I don't know. It truly is one of the more amazing sort of. I'll call it CRISPR is definitely one of the more amazing sort of advancements in emerging therapeutics. It is the lead one at this stage. I'm not trying to say there still isn't a place for ADCs and other sort of therapies, but this is definitely it got published in Science, John. It's amazing work that we did.
Yeah, so like I said, it's very early stages, and I mean the next step for them is to get into animal models to then see what can they do to target extra chromosomes and then address cancers that way. So it'd be
funny. They call it an aneuploidy addiction. What a great I love some of the words they make up there. Like aneuploidy addiction is what they refer to cancer cells having, John. It's great stuff. Sorry to talk over you a little bit there, but it's a really cool article. Yeah.
So then let's move on. I just thought this was worth pulling in. This was because we've gone. We went through COVID where there was a lot of money poured into biotech, etc., and obviously there was all the work we could manage to do out there. But in the last year or so, we've obviously seen that things have really tightened up in the biotech sector in terms of IPOs have dropped off the face of the earth, and there's been obviously so many announcements of companies downsizing or closing doors or things like that. So this was I thought quite a timely article on Endpoints, just saying that despite a summer blockbuster rounds, biotech funding is on track to level off at pre-pandemic norms. So it's really saying that looking back at things, that the pandemic was the freak piece of this. It wasn't a start of a golden age of investment in biotechs, etc. But it has sort of settled down to where we were pre-pandemic, and we are starting to see we are starting to see some IPOs out there, some pretty large, but it's still a very mixed picture in terms of where biotechs are, and so I think the general consensus is it's still going to be like a tough, certainly through 2023. I think, and it's a question where we go into 2024, and I think there's light at the end of the tunnel, but we're still in a tough phase financially for the biotech sector.
Yeah, I think you said it well. They call it a thaw, so it's like a mixed bag, so to speak. But definitely returning to. I don't know if we'll ever hit that. Those pandemic boom years, as they call them, in 2020 and 2021, when money was just pouring into drug development, for COVID specifically. But And a lot of right. We talked about that over the past few years, repurposing of drugs to try to treat COVID or even prevent
Yeah,
it. And now,
yeah,
from the looks of it, if we hadn't had those 2020 and 2021 booming years, we wouldn't even be talking about a mixed bag. It'd be looking
exactly. Yeah, yeah.
good, right? It's kind of right back to where it had been before that, and that's all. To me, it's very encouraging, John, when you see things like 273 million for flagship startup, right? A RNA company for 200 million, a radio pharmaceutical, which is very interesting, for 175 million.
Sure.
I don't know.
Yeah,
It's the on the surface that seems like monopoly money to you and I, but those are healthy startups. But on the flip side, we got to be realistic about what the current temperature is in the market. You still see a bunch of layoffs and some shutting of doors. So
yeah,
it's not quite it's, as they say, a thaw. But
yeah,
we'll stay on top of it and continue to monitor it. But I think 2024 is looking a lot brighter than 2023.
yeah. I mean, I think. I definitely, read a while back about this being like a correction in the market. You're right. Like 2021, there was just so much out there, but it's also, I mean, part of it was the natural. We had companies start up, and a lot of where these, we've seen reductions in workforce. It's been part of the natural progression of drug development, where a drug has had a readout and it has been disappointing. The only difference has been there isn't the investors are being careful with the money, so there hasn't been the money to sustain those companies where they got the disappointing readout. So that then becomes they have to make a hard decision: do they continue, or do they just drop what they're doing? Do they refocus, or where do they go? Because there isn't there's that same. I think the investors have been much more careful about where they're putting money. So, like you say, there have been these big IPOs, but it's obviously like people are thinking that's a company that has legs, and so that maybe there's a lot more, a lot more, maybe more diligence about it, rather than sort of just really propping up companies where things aren't working right, thinking, oh, they may find a way out of that. I think maybe people are being a bit more critical about things.
And I think as to a couple years from now, a lot of them can be they'll resurface, right? That's so, because a lot of it is truly, it's not that the drug failed. It's that you kind of ran out of money, and it's at this like point where you have to either pump more money in it to get it across the line, or maybe it needs a little bit of tweaking. And that's where in the past, maybe money was more readily available. But I think you'll see a lot of, it's just, John, you've been doing this long enough to know a good portion of the drugs that we end up working on were stuff that was shelved for maybe decades before it suddenly someone acquires it or dusts it off and thinks, oh, wait a minute, this could help this disease. So, John, I think that wraps up our news and notes section. We got a pretty interesting topic. It's near and dear to us here at KCAS. That's our autumn conference season, and it is a busy one, John.
Yep.
So we can just dive into the main portion of things. As the main portion today is going to be about our autumn conference season here. And John, first, before we dive into all these conferences, let's just talk about the value of conferences for KCAS. I mean, I think as a company that, we don't have a research department, but this is a big part of what, not just KCAS, I think CROs in general need to invest in. And certainly I feel like we're out there and really active in the conference area. But John, just not only is it a benefit for us to kind of showcase our expertise, but more importantly, it's an ability to present at them. It's an ability to go and have great discussions. Was that missed during the pandemic, despite any sort of virtual stuff. It's much more lively to be in person. The amount of engagements that you can have face to face is tenfold. I don't know, but a lot more than if you were, waiting in a virtual room, which was, we did that for a couple of years. Those were, to me, just terrible. Didn't get a lot of traction versus being in a room or presenting a booth, and they have exhibitor hall times, where people will make their way through and seek you out, or you can seek out individuals. But the benefit is, it's not just the business side. That's the obvious one. It's the science at these that is really some fascinating stuff. And we're kind of, I think of it as, we're fortunate that we're often right at the innovative stage. So a good chunk of the conference is stuff that we might kind of know, but there's always, I put it in this maybe 1 in 5 or 1 in maybe 10 talks where there's something that's just like, wow, wow.
Exactly. Yes.
I don't know what you want to say to that, other than it is like you'll be sitting there, and again, a lot of times there's a conference name, there's a talk name. You'll read the name of it, and you are let down not let down. It's just, oh, that's not as glamorous as the talk name. But every once in a while, and again, I don't know the exact number, but it's not all of them, where you get an attractive topic that you walk in and it delivers. And
Sure.
that is probably the number one. It's like a rush. That's probably an addiction for me when it comes to conferences. But I don't know what else other than some of the obvious things, getting out there, getting to know people. But the amount of, I'll say, technical scientific information passed on at conferences it's just unbelievable. And if you're a novice to a certain area for some of these conferences, we'll talk about that. Then this information is. It's probably, I don't know, it's almost like a crash course in it if you're new to some of the conferences that you go to, and you're either inheriting something or find yourself smack dab in the middle of trying to solve an immunogenicity problem and you're not really sure what that is. Then immunogenicity conference, and we'll talk about a couple of them, those are really, really beneficial for you. But I'll give you just share some thoughts on the value of conferences.
Yeah, so there's a few things. I mean, definitely there's the just getting out and seeing people. We obviously talk to people a lot, but it's on Teams calls, things like that, where you don't really you talk about the business at hand. You don't really talk about anything else, and that's not how you build long-term relationships with people. The best business relationships I've had always included that personal edge where you met up, you get to know about each other's families, interests, and everything else, and you got almost a friendship within that business relationship. So that's an important piece. I think also with us as a supplier, people who are contracting work get comfortable with the labs they're working with. Now, and every lab goes through up and down phases where sometimes you're totally on top of things and everything's going great, and then a couple of key people leave, and then you're not quite doing things and delivering as well as you could. And like I say, all labs go through this. So, if you've got someone who's comfortable with suppliers, they may be dealing with labs that are going through issues, but if they're not attending conferences, they'll be sort of just dealing with those, grinding their teeth and dealing with the fact that the lab that they thought was good isn't really very good anymore. But if you're at a conference and we are there presenting our capabilities, it gives them, it opens their eyes to actually who's out there, and you being there as a potential collaborator. I mean, it's really, really important for us to be not just a presence at a conference, but showing what our expertise is. So that, it might set off a light in someone's head. I mean, I know when I did a talk on GLP-1 analogs, and it's the first time ever I've done a talk, and actually people come to the booth afterwards to ask questions about it in terms of what we could do and what services we had, things like that. So, I mean, those are really important. I do agree the learning piece is essential, and sadly, there's so many conferences that you'd like to go to, and you just don't have you don't have the time, and there's other people also need to go to them. So for big one for us on the mass spec side of things is the American Society of Mass Spec. I haven't been to that in years. That is one where I wouldn't necessarily go expecting to generate business, but that's where I would go to learn. Because there'll be so much stuff, so many sessions, and so many different. It's basically saying where mass spec is likely to go in the future. It's very much a learning conference as opposed to some of the ones that we'll be talking about today. They're much more a blend between business and learning, and it's where you're always assessing the conference about, is this a good one to attend? What are we going to get out of it? What's the benefit to the attendees, etc.
Yeah, and John, I think before we dive into each of the conferences, there's a couple things that can make for lively discussions, right? Like if there are any sort of regulatory guidelines that are new and they're in draft form, and the community is kind of pushing back on some of them. Those can be lively, and I think you know what I'm alluding to. There's also, and I hope I'll be very specific at like WRIB this year. There was a couple different topics that led to some just open, like, not fighting. I don't want to use that word, but just, good. I think it's good, healthy disagreement or good, healthy challenging to the way one a group or one not group, excuse me. I think it's like, think of it as like a hypothesis. There are multiple hypotheses, and just it gets lively. And I think what we're going to see here is there's a lot of immunogenicities falling in that, specifically for some of the emerging technology, emerging drug types, such as cell and gene therapies and others. That becomes a real, like, these conferences help drive how to do that. People are going there to find answers. They go from just, it goes from like, oh, hey, I'm going because I can maybe mingle and learn a little bit. I'm on a mission now.
Sure.
Because my organization and me need some answers, and what better place to go here? Because what you're going to find out is you're at the cusp, and flow's the same way. Spectral flow cytometry, that's the wild, wild west, John. So with that, maybe we jump into the list, but that's why you go to conferences. You go because there's no good guidelines out there, or if there are guidelines, they don't fit to what you're doing. So then you got to go seek out others in the community doing it. And at those meetings, oftentimes this harmonization papers come out of it, good things come out of them. So it is a very powerful, I think, tool for the scientist. And scientists don't love that kind of commit. I think we're a little different, John, that we're very gregarious, right? But that's kind of atypical of a lot of scientists. So it is hard for some of them to go to these things and try and gather information. But it's also like, what better way to grow and get better as a scientist than to push yourself, being introverted to trying to exhibit more gregarious behavior. So, John, maybe we'll just jump into some of the conferences, and first and foremost on that list is
I'll give a bit of a bit of introduction first. Part of the challenge for KCAS is, and it's part of who we are, is obviously we pride ourselves as being a full service bioanalytical organization, but that means that we have our hand in a lot of different pies in terms of analytical technologies. So the range of conferences that we're attending to is pretty wide, and it's obviously there's certain people go to some conferences and that would completely exclude me, for instance. Also, with having a lab in France, then it opens up European conferences far more than we'd necessarily be thinking. Yeah. So in the next 2 months, with one going into December, we're actually planning to attend 10 conferences.
Wow. So you can't do drug development from A to Z and not go to conferences, but look at that. Yeah.
Yes. And so I've never, that tells you that, but like I say, there's two things play into that. Is the diversity of services, and then obviously having two different geographies that we're rooted in. Some were exhibiting it, and some we're more sort of attending. And actually, it might be 9 rather than 10. I see ones was a request. Sorry, I picked that up. Anyway, it's still a lot of conferences. So I just thought it was worth giving that piece, and then I'll let you start and talk through what you want to go with the conferences themselves.
Yeah, I'll just reiterate what just to hammer that home. We have 6 or 7 different service lines. We do drug development from what we think of as A to Z, meaning we can cover your PK analysis on any compound, any type of drug type on the market, and that does lead us to go to a wide range of conferences. So with that, this week we have our Immunogenicity and Bioassay Summit. That, as the name indicates, that's the 15th annual version of that, John. I think I've been to 12 of them or 13. It's a big number. We've got where we'll be exhibiting at that one. Last year we actually presented at it and had a heavy. That's where we opened. We kind of unleashed our magnesium chloride acidification with magnesium chloride poster that helps solve a lot of problems, and it's still a very good tool for the field to help with immunogenicity issues. John, instead of just rattling them off, or do you want me to? How do you want to go? Because we can't spend too much time on each of them. But
I think you just
as the name says, it actually has a bioassay summit component to it, which They kind of added that in like 5 or 6 years ago, which I didn't love because it does throw a little bit of takes the focus a little bit off of immunogenicity and kind of goes into some PK assay. But it's very biologic and large molecule driven, and now it has some elements of like cell and gene therapies and other things, but there are now specific immunogenicity conferences for that. So that one it's supposed to be a little more focused, but it's always going to have some peripheral stuff. But it is focused on biologics. And with that maybe we'll talk about the next one on the list. It's the meeting on the Mesa in Carlsbad, California. I have been to that one once, John. That one's a pretty eclectic meeting. It's got a little bit more of the financial end to it, but it has a heavy, that's going to be heavy on all the emerging sort of technologies. So it's got a heavy, let's just say, cell-based and gene-based and a lot of cell-based needs. And, maybe you can jump in before I go into the next one.
I mean, to me, I don't know. With these, obviously, these don't impact my life.
Yeah. No, so, but that meeting on the Mesa one, so the Immunogenicity Bioassay Summit is maybe 600 or 700 people. It's not, or less. It's 300, 400, but they're all there. It's heavy scientist driven.
Sure. Yeah.
The meeting on the Mesa is like over 1,000, and it's going to be heavy financially driven. See how that works?
It's a question. Do we know any of the attendees of some of these? That would be an interesting thing to raise. And, I
know our competitors more than so.
Okay.
And I'm not mentioning them, but no, like the immunogenicity and it's always dominated by the pharma.
Sure.
The large pharma is and biotech is dominating the immunogenicity. And then companies like 4D Molecular, those that are getting into the cell and gene, anybody starting to get into the CRISPR space, starting to get in, or not starting, but have been in that space, heavy in the gene therapy space, heavy in this, they're going to go to these immunogenicity ones. The meeting on the Mesa is a who's who of companies. It's definitely going to be incredibly diverse, and a lot of CROs are attending, and competitive wise, it's got a little bit more of a CMC component to it. That's the other challenge here, meaning, we're bioanalysis, so we pick up after that sort of GMP CDMO type of stuff. So there's some of that at the meeting on the Mesa, and we're not going to get through this list. So we'll just.
I also wonder I actually meant people from KCAS who were attending.
Oh, sorry. Well, at the Immunogenicity Bioassay Summit, we've got Lindsay Rutherford, our scientific advisor for ligand binding assays, and Tom Esposito. I was going to call him Esposito, but Tom Esposito, who's our business development director for California area or outside of the Bay Area, will be there as well. They're exhibiting, and then at the meeting on the Mesa, we'll have our chief commercial officer there. Jeff Goddard will be at.
Okay, so yeah, you're right. We can't spend a long time on all these. You got the 3 in Europe then in mid-October, Festival of Biologics, which we're exhibiting, and I know of at least one customer from the US who's going to be there, so our Lyon team will be primed to see them. I assume they'll also be at the World Vaccine Congress and GMP Symposium in Paris, France. World Vaccine Congress is in Barcelona. The Festival of Biologics is in Basel, Switzerland, so it would be nice set time for that. And then the next big one for the US contingent is really AAPS PharmSci 2023 in Orlando, Florida, from October 22nd to October 25th.
Yeah,
and I'll let you talk on that.
Yeah, just to quickly recap, though, the World Vaccine Conference in Barcelona, right? There's a Paris one, the GMP Symposium, and there's a Festival of Biologics Basel. I haven't been to any of those, but I have been to like the World Vaccine Congress in the U.S. It's a great meeting. That's an area that, John, I think if someone said, where could you see KCAS going? It's we support some vaccine work, but I think that's really an area where if you ask me where we invest some dollars in, say, 2025, it would be in that area.
Sure.
I think that's something that's coming. And then obviously GMP, we kind of pick up as people get into that pilot before they go to make their true GMP grade. That's kind of when we integrate. And then the Festival of Biologics, the US one, is massive. So I'm not familiar with the European version of it, but it's about biologics in particular. But John, with that, I thought it'd be important to touch on those a little bit.
Sure.
For all of our active biomarker or our Lyon site, we'll be going to that. But then, as you said, the big one coming up is the AAPS one, and that is in Orlando. It's the 22nd through 25th. John, I'm so active in the AAPS community. I was going to try to squeeze in a critical reagents talk of some sort, but fell a little bit short. But I'll just start with, there is a dinner as a leader. I'm a leader of a focus group, and that is the critical reagents focus group. And so we have this dinner there, and I'll have to give a little update on what we've been doing. And it's a nice honor. Not that we're being honored, but all the volunteer time over the last year, we get a little bit of a reward with a dinner, and we get to hear about how others are doing. And it's really a cool little thing that I'll get to attend and see other leaders in the community and get to sample from what they're doing, so that, the whole goal is to produce something that is meaningful for the community. Specifically, this one is critical reagents, but I'm on the PK group. I'm on a NAB assay one. There's a publication coming out for that. I'm on the PCR one. That's a second year, although Carrie Vidal is probably more active in that group than I am. I'm actually on like a data management one. I'm on a liquid biopsy one. I mean, the list goes on and on. It's hard to be active in all of them, but really the two big ones for me are the critical reagents and the PK. But that meeting's very, very big. But there is a good section, because it has a lot of pharmaceutical alumni, right? Pharmacist alumni. That's not really germane to us. And it has some GMP components to it. But the bioanalysis section of it is rather big, and it's definitely becoming more and more prominent there, because it's an area where people are struggling. I think bioanalysis is a contact sport. You hear me say that sometime. It is definitely one of the more challenging areas in all of drug development. In fact, I would say it's arguably the most challenging. We're at the tip of the spear when it comes to, finding a way to measure your drug in a biological matrix, or trying to figure out the mechanism of action of your drug in some sort of sample. Not easy tasks. And I'm not trying to say GMP work or true CDMO work is easy. I'm not belittling that in any way. I'm just saying, when you add matrix and it goes, okay, I got to look at normal donor serum and plasma or matrix, then I got to look at disease state. And if I want to go into multiple disease states, all of that has to factor in. And then, not even mentioning some of the challenges with looking at pharmacodynamic and biomarker assays. All of that's addressed here. It's a wonderful meeting, John. Yeah. I'll take a small breath, but you can tell me I'm pretty excited about this year's meeting. It'll be a nice one.
So we'll be exhibiting there, and then I don't know how many people are attending.
I think we have 7 going this year.
A mix of business development people and then more scientific focused people as well.
Yeah, I think we have 7 or 8 attendees this year, which is down. In years past, we've had like 13 or 14. It's just it's something that we've exhibited at in the 10 years I've been here, or 11th now. This will be the 11th, but I think we've been exhibiting at that for over 20 years, as far as I know. Sure. I guess it's something to check. I think we've been doing it for quite a long time. With that, John, the next one I'll touch on, it's called the Metro Flow 2023 annual meeting. It's in New York City, and it's as the name implies, it's flow cytometry based. So our SHIP team, the former Flowmetric team, we call it SHIP, Springhouse Innovation Park team. They'll have a presence there. Brian and I believe Jonathan, the Mid-Atlantic rep, will be attending there. And it's I don't want to call it a small show, because I think it has close to 1,000 people attending to it. It's the first I've heard of it, but it's flow heavy. And as I kind of alluded to earlier, the spectral flow space is really, really open in terms of some of the challenges that come with trying to do 40 parameter flow cytometry or 60 parameter flow cytometry. And we could do a whole podcast on that in and of itself, or potentially a whole series, maybe even a separate podcast for flow, because it's such a dense topic. There's so much involved with, and just the evolution of flow to go from, they were dabbling in 4 color to 10 color, 10 color to 20 color, and it kind of staggered. There were some instruments that could do more than 20, but they were really researchy instruments. And then spectral kind of splashed onto the market a couple years ago, and it's great. But now we're having challenges with not only how to get the fluorochromes to play nice on the sample upfront piece, on the sample analysis piece, it's the data. I mean, wow, artificial intelligence, and they call it unmixing of the data, how to control for that, all still very, very challenging things that we can unlock some of those challenges, but this Metro Flow meeting is an area to go and learn more about what people are doing to as I said, kind of unlock those challenges. And then, John, maybe the next one we'll give you a chance, because I do think we do have some feedback. We want to talk, like a question from our audience from last podcast. So I want to be cognizant of time. Tell us about EBF, because I think
actually, a couple of things. I mean, we had the chat before this, and our producer will actually be in Australia for a AusBio meeting at the beginning of November, and I assume there'll be a chance to see what is out there in Australia for us. And then I'll jump in terms of where I will be attending the European Bioanalysis Forum in Barcelona in mid-November. I think it's about 15th or 16th year, and again, I've been to most of them. This year we have two podium presentations. So we're doing one on the analysis of urea, so a small non-polar biomarker, which analytically is very challenging, and then another will be talking about use of hybrid and some of the issues that we encounter with hybrid LCMS. And this isn't just an LCMS meeting. It's around, I think, 600-700 people. It's very scientifically intense. They'll have sessions that are for all attendees. Then they will actually do dual track, so ligand binding and LCMS. I'm sure there'll be flow and stuff in there too. It's a short meeting but intense in that, there's a lot of talks when it comes to the exhibition phase. That's where you have lunch, and so people are having lunch in amongst the exhibitors, so it's actually a good chance to talk. There'll be evening mixers, so there's more chance to talk with other attendees. It's a really good meeting in terms of talking to European counterparts, and there's usually a good US component. I'm sure this will be a good year for US attendees, because EBF tends to float a bit, and sometimes it coincides with Thanksgiving. That's when the US attendees go down. This year it's the week ahead of Thanksgiving, so yeah, I would expect to see quite a few people over from the US too. So yeah, a really good meeting.
Yeah, it's like their WRIB, right, John? It's like their RIB, the WRIB exactly. Of Europe. It is probably the number one.
Yep, it is in Europe. Yes,
development conference in Europe. Sorry,
and we'll be exhibiting there. So we just picked our booth last week. We know where we're going to be. So yeah, it's because again, it's with us. And there'll be I think there's 3 going over from the US from KCAS, and then we have contingent from active biomarkers as well. So our new site head Musan will be there, Christine Bain, and then Magali Roche. I'm not sure who else will be attending from active, but yeah, it's a good. If we had to choose one meeting in Europe, that's the one we would always go for. It's excellent.
Who's the? Did you say Hassan? Who's the new?
Musan.
Okay.
Musan Ufir is going to be there.
Yeah, he's the new head of our Lyon site. Yes, we welcome him with open arms. Yeah, great hire for us.
So hopefully then at some point he'll be a guest on this podcast, and we can talk to him more about his philosophy and vision for Lyon.
Yeah.
And then the last on the list, and again, I don't know, is the LNP Immunogenicity and Toxicology Summit in Boston, Massachusetts in December. And I don't know much about that one.
Yeah, this is I think it's the 3rd year for this one, and immunogenicity is all the rage. Last quarter we went to the cell and gene therapy immunogenicity. There's an immunogenicity bioassay summit. There's an immunogenicity of biotherapeutics summit. That's a separate immunogenicity of biotherapeutics is a separate conference, and then there's this LNP immunogenicity. So I think there's 4 or 5 specific. And so this one's I think it's in its 2nd or 3rd year. And John, there are some real not challenges. LNPs you got to unpack them. That's the best way to say it is, do I develop my immunogenicity against the full intact LNP? Do I do it against the material? It's got some complexity to how to design and how to tackle LNPs. And this conference focuses on it. And it also has that toxicology component to it. So it does look at safety, and the word immunogenicity is a dense word in my opinion, because some people vaccine it's wanted immunogenicity, and then the rest of certainly biologics, it's typically unwanted.
Yes,
Although,
yes.
John, as we know, most of the drugs that are, a lot of the biologics or even cell and gene therapies that are highly effective have immunogenicity. It's a dual-edged sword. It's tough to kill a cancer cell without having some augmentation of one's immune system, and that becomes a safety issue. So there's that. The reason why there are so many of these highly focused immunogenicity conferences is because of the challenges and the
Sure.
desire to get together and see what others are doing, share that information in real time is what these conferences allow for. So I think that's the third or fourth year of that one. I'm not quite sure. I haven't been to that one. I don't know a tremendous amount about it. I think it's our first time being there, but we're actively working on 3 to 5 immunogenicity assays for LMPs, in addition to the equally number of on your side of the fence, John, looking at them by mass spec or something.
Sure. Yeah, exactly. Yes.
So it's definitely an area of expertise. And so, John, that was a fun main topic. Before we dive into what we're doing, maybe we'll just jump right into the feedback and question that we have. Does that sound good to you, John?
Sure, absolutely. Let's go for it. Basically, in response to episode number 68, which we did on cell and gene therapy with Carrie Villadal, Bert says, great podcast. I find myself getting more and more involved in bioanalytical assays for proteins. My 35 years of experience has been in small molecule chemistries. In today's changing world of pharma development, I need to gain more understanding of the assays for proteins. Have you ever done a podcast for beginners like Bioassay 101? So I've just had that.
We have, John. Actually, I thought we had done it very early on, and
yes,
our producer is letting us know it was episode 5. I do recall it was like Bioassay 101. I think we did or everything you wanted to know about. You remember I did some training years ago internally, and I think we adopted it to a podcast. It was like everything you ever wanted to know about PK assays for biologics, but we're afraid to ask, and everything. I don't know if you remember all of that, but that we did an episode. It was really a 3-part series that started with episode 5. So we've kind of dug in, but that's years ago, John.
Yeah,
so I think it's a great reboot. I mean,
sure,
that's near and dear to my heart. So I always love taking a topic that is going to be something. But maybe we even try to bring in Shek Kane for that one, and maybe even some of our more seasoned method development scientists, so that they can break down a little bit of the mindset behind a checkerboard assay, for instance, because that would be a great little first towards Bioassay 101.
And complement that with someone from the hybrid side. If we really focus on proteins, then you could do a mix, and we can cover it that way. I mean,
maybe a series. I think we're on to maybe having part 1 would be LBA kind of based, focused, and then part 2 would be the hybrid. Because you're right, the bioassay space has expanded to hybrid mass spec. It's definitely a great tool to be using, and more than just a tool, it is a solution. It is a platform that can be selected as your go-to platform for biologics. Although the ligand binding assay still. That's a hot topic, right, John? Great question, Bert. Really appreciate that, and we'll get that on the docket, and it's definitely something we'll tackle in 2023.
I mean, another thing I would say is keep an eye out for webinars that come from us, because we are talking about doing a webinar in early December on small molecule biomarkers. Collaborating with SciEx on that one, and then they'll actually. We hope that we can do more behind that in terms of other LCMS techniques, and that would obviously bring in hybrids.
All right, John.
That kind of thing too.
Real fast. Tell me what you got going on in your world. Anything going on this weekend? Anything you want to share with our audience?
So it's going to be really, really quick. I'll have to be really quick. Our life has been somewhat hectic for about the past 6 weeks or so, because obviously my daughter got married in 2nd of September, which was honestly just a brilliant, brilliant weekend. We had family over. The weather was fantastic on the day. It was just a really excellent day. Food was great. Everyone had a wonderful time, and to be honest, we were basically glowing in our success of my wife's organisation. She did a fantastic job pulling it all together, and so the last 4 weeks have still been, I can't believe that went so well. It's funny that, but this coming up weekend, we're actually going to another wedding of one of my eldest son's friends from college is getting married to a girl who actually lives close by us in the Finger Lakes region. So we have a wedding to go to this coming weekend. And at the moment, it's like my wife and I are juggling different travel schedules, and our October, we hope, will be it'll be a lot of work travel. We'll hope October will be relatively quiet, but then November becomes I'm traveling back to the UK to see friends and family. So
we are in very different spots in our lives, John. We're in very different spots. I have a son who's in high school, so he's a senior, and there's a lot of events that I'm doing that I find myself at for him that are not nearly as glamorous as what you're talking about. But, high school football games are all the rage. But I'll just share, the thing that I want to share with the audience is, if you don't know, I have a uni pizza oven, and I feel like I've mastered that. But I have a big green egg. And so I had a bunch of badges, as my son kind of talks about it. I've gotten I got my brisket badge. I got my pork shoulder badge. I had been struggling with the chicken wings, but John, this past weekend, I just nailed them. I nailed the chicken wings, and then I've moved on to some vegetables. So I actually did a spaghetti squash, which was, I can't believe how good it came out. And then I grilled some zucchini as well as some summer squash on there. And I can't go a podcast without sharing some of what I'm doing with my food. But I don't have anything really that big going on in my life other than keeping up with my son's schedule. He had some homecoming dances and all these things that I'm happy to go and see him growing up and before my very eyes. But other than that, it's watching some football and really enjoying that. But John, with that, we'll say goodbye to our audience, and we're excited to maybe do a bioassay, but stay tuned on the next podcast.
Exactly. Yes. Thanks a lot. Look forward to next time.